Okay, so let's start. Sorry for the delay, guys. It's a technical issue. Okay, first of all, we are having the structure, and then we are moving to the essay, and finally, if we have time, we are doing the essays. Okay. Now you will have 4 structures in your final exam. So the first one mainly targeting the foundation and some of the animal diversity part. So I'm going to do the first structure now. So the first thing is state 3 characteristics of first formed organisms on Earth. So you all know the first form organisms uh, should be a prokaryotic and a anaerobic because there's no oxygen on Earth. And the third thing is heterotrophic because there are no any other plants to supply food. So it should be heterotrophic, anaerobic, and prokaryotic. Heterotrophic, anaerobic, and prokaryotic. So these are the 3 things they have mentioned in your resource book. So if you write all those things, you will get two marks. Or better if you write all 3 or any 2. The next question is, what are the environmental problems that arise due to overexploitation of natural resources? These are the questions I took from your resource book. The earlier part that is not so important. The students think that is not so important, but they are asking questions from that. So the first one is environmental pollution. The second one is loss of biodiversity. And the third one is desertification. Okay, if we use the natural resources in an extreme way, we'll have all these problems. So the next question is, one of the characteristics features of living organisms is adaptation. What is known as adaptation? So I'm not going to tell you the definition. You have the own definition on your resource book. So it is a peculiarity of structure, physiology, or behavior of that promotes the likelihood of an organism's survival and reproduction in a particular environment. The key words are, you need to say that it's the structure, physiology, or behavior that promotes the likelihood of organisms, survival, and reproduction. The survival, reproduction words are the key words. And the structure, physiology, and behavior, you need to mention all those 3 words in order to get the full marks. Okay, if you have any doubts, you can unmute yourself and let me know. The fourth question is, mention 4 major properties of water to maintain life on Earth. These are the easy questions I took from your resource book, but they are asking those foundation questions as well. So the major properties of water to maintain life on Earth is the cohesive behavior, ability to moderate the temperature, and expansion upon freezing, and the process as a solvent. So these are the 4 properties of water that is essential to maintain the life on Earth. So the next question is, what are the interactions between the amino acids of the tertiary structure of protein? So you know there are 4 structures of protein: primary, secondary, tertiary, and the quaternary structures. So the primary structure is just a polypeptide of amino acids. So there are amino acids polypeptides. If two polypeptides are arranged together, we call it secondary amino acids. So 2 or more. The tertiary amino acid is 2 or more polypeptides are arranged together. So if there are a lot of polypeptides like this arranged together, they might have some sort of interactions between them. So the first one is the hydrogen bonds. You know the hydrogen bonds. You can write like this in your examinations like H bonds. You need to write hydrogen bonds. So the second one is disulfide bonds. Disulfide bonds in the sense there are bonds between the sulfide atoms. So it is called disulfide bonds. And the ionic bonds, as you always know, the ionic bonds between plus and minus, there might be interaction. And the last one is the van der Waals interaction. So you can call it as hydrophobic. Okay, hydrophobic interactions. So if you get all those things correct, you can have 2 marks for these, 2 marks for these, 2 for these, and 4 for these, and 4 for these. So for the part A, we have the marking scheme like this. So if you have any doubts, can you unmute yourself and ask me? This is a so easy question, so I'm going to move to the next part now. Can someone say anything? Guys, are you there? Do you have any doubts on these things? So I take it as yes, that I'm moving to the next part. So the next question is state 3 types of RNA and mention the functions of each. So you know mRNA, which is known as messenger RNA, tRNA, transfer RNA, and the rRNA. So you all know in the cell we have the nucleus. In the nucleus we have the DNA like this. So the DNA as the nucleotide copy of everything this body needs to do. So the messenger RNA comes and copy those things, and it goes out and give the information to the a cell or what protein needs to be produced. So the messenger RNA, as the name implies, it copies the message from the DNA and come outside and give the message to the workers who are there to do the work. So the messenger, the function of the messenger RNA is copies the genetic information stored in DNA as a sequence of nitrogen base. So you need to clearly write the functions of mRNA. One function, which is it copies the genetic information from the DNA. That's enough. The next one is the tRNA. The tRNA is the one who transfers the amino acids needs to build a protein. So if you're building a protein, you need various kinds of amino acids. So let's take, we have alanine and cysteine, some sort of thing. So the tRNA is the one who brings those guys to the site of protein synthesis. So if this is the site of protein synthesis, this guy brings the amino acids in the place to build a protein chain. So the next one is the ribosomal RNA, which is the RNA present in the ribosome, and it provides a site to the polypeptide assembly during the protein synthesis. So that's it. So the next question is, the endoplasmic reticulum is a network of internal membranes forming flattened or tubular sacs. So you know there are endoplasmic reticulums like this. With the nucleus, you have some sort of endoplasmic reticulum like these bags and everything. They might contain ribosomes, or they might not contain any ribosomes like this. So if they contain the ribosomes, we call it as rough endoplasmic reticulum. If it doesn't contain the ribosomes, we call it as smooth endoplasmic reticulum. You all know that. So there are particular functions for both those endoplasmic reticulum because the one guy is containing the ribosome. So we saw that ribosome is involved in protein synthesis. So the rough ER or rough endoplasmic reticulum is a main part of protein synthesis. And it can also synthesize glycoproteins. Glycoproteins, you know, which is a combination of carbohydrates and proteins. And the next thing is it can produce vesicles. So after they produce the proteins, so let's say this is the proteins, so they form a vesicle like thing. This is the ER. They form a vesicle and transport to the places where the proteins meet. So it forms the vesicles and it facilitates the growth of the bone membrane. What do you mean by facilitating the growth of bone membrane? So if this is a cell, this is a cell has a cell membrane, which is a double layer. So, which is a double layer. Um, it has some, it needs protein, like the transmembrane protein, uh, surface protein. There are so many proteins. So, in order to make a cell membrane, these endoplasmic reticulum will send the proteins to the cell membrane as well. So if you write any two of those fours, you will get 1 mark. And the functions of the smooth endoplasmic reticulum is to synthesize lipids. So if this guys, if the rough ER synthesize the protein, the smooth ER synthesize the lipids. What sort of lipids are there? There are oils, steroids, and the phospholipids kind of things. And it involves in the metabolism of carbohydrates. And it also produces vesicles because it also producing some sort of biomolecules, and it also produces the vesicles. And it is mainly involved in detoxification, and it stores calcium ions. Okay, so if you guys have any doubt, please ask me. So to summarize these things, the rough ER involves in protein synthesis, the smooth ER involves in the lipid synthesis. Both are doing some sort of production, and they both are packaging things in vesicles, right? So, and the main thing, the smooth ER is involved in the detoxification and stores the calcium ion. We'll move to the next question. The cytokinesis is the division of cytoplasm at the end of telophase. Mention the structures that form in plant and animal cells during cytokinesis. So, you know, the cytokinesis is the division of cytoplasm. If two cells are dividing, so here we need to divide, at the end, we need to divide those two cells in order to get two different daughter cells. So the process is same. It is called the cytokinesis, but there are two different structures formed in animals and plant cells. So in animal cells, we have the cleavage furrow, and in the plant cells, we have the cell plate. These are simple questions. And the next question is, what are the main steps of aerobic respiration? Mention the location of each of them takes place. So there are 4 steps of aerobic respiration. The first one is glycolysis. So where does the glycolysis happen? It happens in the cytoplasm of the cell. The second one is pyruvate oxidation. It's a linking stage between the glycolysis and the Krebs cycle, but we need to mention all those 4 steps because it was in your resource book. Pyruvate oxidation and the Krebs cycles both happens in the matrix of mitochondria. So you need to mention clearly as matrix of mitochondria. If so, if you only mention the matrix, you will not get any marks. If you mention the mitochondria also, you will not get any marks. The last step is the electron transport chain. Sorry for writing in the shortened form. Electron transport chain, we need to write in the full form. So it happens in the inner membrane. Inner membrane, or you call it as cisternae of mitochondria. But you need to mention clearly as the inner membrane of mitochondria or cisternae of mitochondria. Okay, so that's why these two have the specific names. If you write only the matrix or the inner membrane, you will not get any marks. So the four main steps are glycolysis, pyruvate oxidation, Krebs cycles, and electron transport chain. And it happens in cytoplasm, matrix of mitochondria, and inner membrane of mitochondria, respectively. So the next question is, what are the two types of cofactors? So there are two types of cofactors. You all have studied in the enzymes, I guess. So two types are organic cofactors, and the inorganic cofactors. Organic cofactors are NAD plus, FAD plus, and the biotin. Sorry, no plus mark is there. NAD, FAD, and biotin. So these are the organic cofactors. Inorganic cofactors, you can say the metal ions, ZN2 plus, Fe2 plus, or Cu2 plus. Okay, for the organic cofactors, you can write as coenzymes as well. Guys, if you have any doubts, please unmute yourself and ask me, because I'm not able to see the chats in the middle. Okay, I'm moving to the part C now. State the functions of the following structures in animals. So this is the part of the diversity. So the mantle and radula, where can you see mantle and radula? It can be seen in the mollusks, right? Okay, mantle is a covering of the body, covering of the shell. So if this is the shell, it is the one who produces the shell. So the function of mantle is to synthesize the shell. So the radula also you can see in the mollusks. So it is a structure in the mouth. It is like a razor blade. You can see the razor blade like thing inside their mouth. So it is used to feeding. And the mantle is used to secrete the shell. Right? The parapodia can be seen in the annelida, right? The annelida, the polychaetes of annelida, like it will be some sort of this structure. So the parapodia can be seen in the polychaetes of the annelids, and most of its we know is Nereis, right? So the Nereis has the parapodia, and it is used for the locomotion and the respiration. And the last thing is the swim bladder. You all know it can be seen in the fishes. It can be seen in the fishes, and it's used to the control of buoyancy. Guys, can you hear me? The swim bladder you can see in fishes. It is used for control of buoyancy. Right, the fish needs to be swimmed, and it cannot be like sink under the water, so it needs a kind of buoyancy. So the fishes have the swim bladder. The next thing is state two features seen in heterocircle caudal fin that could be used to distinguish it from a homocircle caudal fin. So the heterocircle caudal fin is like this. Like you have this. It has two. It has the fins which are unsymmetrically divided. So the features are the upper lobe. The upper lobe. This is the upper lobe. The upper lobe is larger than the lower lobe. The first feature is upper lobe is larger than lower lobe. The second feature is the vertebral canal. The vertebral canal from the brain will extend to the lower lobe. It will go like this. So that is a feature. So the vertebral canal extends to the upper lobe. So the next question is, name the hormones that contribute to the following functions. Functions in a phototrophism and rapidotrophism. You know, it's a growth towards light and the growth towards the gravity. So it's the growth of the upper part and it's the growth of the root side. So it is facilitated by the hormone called auxin. The next thing is promote ripening of many types of fruits. We use ethylene. To promote stomata closure during growth stress is the abscisic acid. And to stimulate pollen tube growth is the gibberellin. So these things you need to be by heart. So that's it for my part. Do you have any doubts or questions to be clarified?
Okay, I hope you all can hear me. Then let's start. So now we are going with the third question, animal form and function. So the thing is, what I want all of you all to do is listen to me carefully, and I will give you all time to write down stuff and ask me questions. At the end of, I will let you all know when you all can ask me questions and write down notes. I'll give you all a small amount of time. But when I'm explaining, everyone make sure that they listen carefully, okay? So let's start. First thing, what are the ventricles in the brain? So when it comes to ventricles, the first thing we should know is, okay, so what is the definition for ventricles? That is a clear thing from the resource book itself, you all should know. So they are irregular shaped cavities. So they are irregular, if, wait, I'll write it clearly. So they are irregular shaped cavities formed by the central canal, and that is filled with cerebrospinal fluid in the brain. Okay, so then when looking at that, so the first thing we have to know is the embryo. So during the embryo, so in the embryonic state, what's the shape of the human? It is the shape of a comma. So a comma in the sense, if I draw the comma like this, it will be, I mean, something like this. So it's the shape of the human, the embryo. And so then there will be a hollow dorsal cord like this. Okay, so there will be a hollow dorsal nerve cord like this. The hollow dorsal nerve cord in the anterior portion will enlarge and form the brain. The anterior portion will enlarge and form the brain, while this posterior portion will remain as the spinal cord. So that's the normal way it is. So this brain part only, so the brain part only will form into these irregular shaped cavities called the ventricles. So that's the first thing. So how many ventricles are there? So that's the next question. You all can, I'll show you the next question. So the definition for ventricles are irregular shaped cavities formed by the central canal filled with cerebrospinal fluid in the brain. How many ventricles are there in the human brain? There are 4 ventricles in the human brain. So what are these 4 again? So 4 in the sense, I'll show you back again. So here, when we talk about the brain, okay, so the brain has 4 ventricles. And so when we look at how the ventricles are distributed in the brain, first let's look at what the brain consists of. The brain consists of the forebrain, the midbrain, and the hindbrain. So these ventricles are distributed how? 3 ventricles go to the forebrain, 1 ventricle goes to the hindbrain. So remember that also. Next, what are the structures containing cerebrospinal fluid in human? So that is, very easy here. The word itself gives the answer, cerebrospinal. So cerebro and spinal means, so here it comes the spinal cord. And cerebro means the head region, no cerebral region, the head. So here it comes the, what, ventricles in the brain. So ventricles. So then these are the 2 structures, ventricles and spinal cord. Okay, so the spinal cord, more specifically, you can say the central canal in the spinal cord. So central canal in the spinal cord. I hope I'm writing it clearly. So central canal in the spinal cord. Then next, so indicate the function of the above mentioned fluid other than the maintenance of regular pressure. So normally any fluid, okay, in the body, whether, so the fluid which we are very familiar with is blood. So almost every fluid, that's the same function. The main function of every fluid in the body is what? To transport, circulate nutrients. That's the main function. Circulation is the main function of anything. Any fluid, mostly any fluid in the body, purpose is circulation. Circulation means helping transport it, exchange it. That's the purpose. So here, circulation of what? Circulation of nutrients and hormones. Circulation of nutrients and hormones. So these are directly from the resource book. And so these are very interesting parts. You all can study it and remember it for forever. So circulation of nutrients and hormones, because the brain has several organs. So the brain itself is the main organ. It has a part called hypothalamus that produces several hormones. So it needs this cerebrospinal fluid to circulate nutrients and hormones. So circulate nutrients and hormones. Then again, remove waste products. Same as the blood. Blood also transports waste, carries waste products. So again, remove waste products. And then finally, acts as a shock absorber. Acts as a shock absorber. So what else acts as a shock absorber in our body? Another fluid. If you remember, between in the lungs also, there is something called pleural fluid. So that also acts as shock absorbers. So these are the 3 other functions that you can write for the 3 part B. Okay. Then next, okay. So these 3 A, 1, 2, 3. You can take a screenshot of the screen if you want. And if you are free to ask any questions if you all have. So please feel free to ask any questions. You can ask by unmuting or by putting it in the chat box. If you all understood, just say that you all understood, so I know that you all understood. Okay. One person put understood, so that's fine. Take a screenshot, okay? Take a screenshot, and even this, if you all want, you can take a screenshot of this also. Okay, then moving on. So the next thing is, what is the reason for hyperpolarization? So now the brain is over. Okay, now the next is the neurons. What is the reason for the hyperpolarization? So underline it. Hyperpolarization taking place finally when an action potential is generated. So now the question comes, what is an action potential? What is hyperpolarization? Okay, so the first thing we have to know is, when we talk of an action potential, so we'll go here. This is the neurons. Okay, so we are learning about nerves. When we talk of an action potential, AP, it consists of what? What does action potential consist of? It consists of depolarization. So depolarization. Then it consists of repolarization. Then it consists of hyperpolarization. Then only you get this refractory period. Okay, so now what is this action potential? Action potential is simply how you make, now in a neuron, so now think this is the cell membrane of a neuron. How do you make these electric impulses go? How do you make electric impulses flow? So that is only action potential, okay? So then to do action potential, to make the electrical impulses flow, you have to use these 4 steps. These are the 4 steps. So firstly, what is this? So I will give you a quick recap. Depolarization means, remember, in a cell membrane, usually this is the cell membrane of a nerve, a neuron. Always inside is negative. Negative what? 70 millivolts. So this is the normal way. Okay. So this is the resting membrane potential. Depolarization means change in this polarity. Polarity means a charge difference. So minus 70. That means compared to outside, inside is minus 70, right? So depolarization is making a change in this voltage difference. So how do you make a change? So this is by the action of the sodium. So then what happens here? Sodium voltage gated channels or voltage gated channels open, and this results in the sodium sodium inflow. So sodium ions come in and makes it positive up to what? Plus 30. So this is the first step, depolarization. Then what is repolarization? Repolarization means now you made a change. Earlier you are normally minus 70, now it's plus 30, and bringing it back to where it was. Repolarization, bringing it back. So then that needs K plus outflow. So here sodium ions come inside, here K plus ions should go out. Okay, so then that will only bring it. So if K plus ions are going out, again inside will try to come back to its normal minus 70. But what happens? These K plus ions the outflow of K plus ions is prolonged. That means continuous outflow. You didn't stop the outflow properly. So then that will make the inside, usually it should be minus 70, right? It will make it more negative, say minus 80, for example. So that is hyperpolarization, making it more negative. And refractive period is what the question is asking for. So we'll check what this refractive period is. So the refractive period, so this is again, when it comes to biology, it's definitions only, which you all have to memorize. Okay, so what is the refractive period? No, that was not the question. What is the reason for hyperpolarization taking place finally? So what's the reason for hyperpolarization taking place? So when the answer is, so when the sodium ions, so I'll show you here, sodium channels. Okay, so here, first depolarization happened, right? Sodium ions inflow. By how? Sodium ion channels that opened here. There were sodium ion channels here that opened. Okay, so these were sodium ion channels. They opened and allowed sodium to come in. So here that sodium ion channels closed and potassium ion. So this is the sodium ion channels. These closed and potassium ion channels opened. So here what happened? Potassium ion channel that opened remained open for a long time. That's why prolonged. So what's the answer? Sodium ion channels were closed, and potassium ion channels are opened. And the opened potassium ion channels, so they resulted in K plus potassium ions outflux, which make these open potassium ion channels remained open for a longer period of time, or prolonged potassium outflux was facilitated. Therefore, this makes the inside of the cell more negative. So that's all you have to say. I hope you understood. Sodium ion channels closed, potassium ion channels opened. Potassium ion channels remained open for a long time, or prolonged opening, and that results the inside of the cell becoming more negative. Okay, now, so that was the first question. Then the next is, what is the reason for the formation of refractory period? Okay, so let's see what's refractory period. So now what happens here? Refractory period, or the purpose of refractory period is, what is the reason? Why do we have a refractory period? This is to prevent what? To prevent the reverse conduction. To prevent reverse conduction of nerve impulses in an axon or in a neuron. Axon means neuron, okay? A part of a nerve. So prevent the reverse conduction. So usually this is what happens now. Say this is the nerve. This is the neuron cell membrane, okay? Cell membrane of the nerve. So cell membranes are present both. So cell membrane on both sides. So then this is the cytosol. Okay. So here what happens is, so usually when they say an action potential is happening here, so say here it becomes plus 30 inside. Action potential means it includes all these 4 steps. So here it becomes plus 30 inside. So then this plus 30, okay, it becomes plus 30 inside, and the other areas remain in rest, minus 70, minus 70. Okay. So now this plus 30 generated inside is due to what? Due to sodium ions coming in, right? These sodium ions spread these in both directions, in forward and backward directions of where the action potential is taking place. And now this sodium ion that came forward here and here, they will again try to create another action potential. That's the normal way. Okay. It will try to create an action potential. So you call this mechanism how? Local circuits. This is called local circuits. So via local circuits, action potentials are propelled forward. helping an action potential that started here to come here. So now normally if an action potential or if a nerve impulse wants to go from one end to another end, it should go in a unidirection. No? If it's going this way, it should go in this direction. So it can't go back from where it started. It can't come back to where it started. No, it should go in a unidirection from the receptors to the brain and from the brain to the effectors. So that is the normal route it should go. It can't go in any other way. So now what happens here is because every nerve impulse, every action potential has a refractory period at the end. A refractory period only prevents the reverse conduction. So what happens here? When the sodium ions try to go forward and backwards, this region already underwent an action potential. So it is in the refractory period right now. If it is in the refractory period, it can't undergo another action potential. So therefore, even though the sodium ions come back, it can't make this side get activated. So only the sodium ion that went front only can generate another action potential here. Then you can see always action potential is moving how? Forwards. Forwards means in one direction. So that is the main purpose. So what is the refractory period? It prevents the reverse conduction of what do you call prevents the reverse conduction of nerve impulses in an axon. What is the reason? If you want to know, remember that there were sodium ion channels right here that always result in the depolarization. Remember that the sodium ion channels which are needed for depolarization, that gets inactivated. So that gets inactivated. So that's why you can't do a reverse conduction. So remember that. Okay. And next, so let's see the next question. Okay, before going to the next question, I think, yeah, I will stop it here. You all can ask a doubt if you have any, and take a screenshot of this if you need to. Can we get the PDF of the tube? Wasn't it sent? Okay, so I'll send that later. Can we get the PDF? I'll send it a bit late after I'm done. Until that, take screenshots and keep it, and then you can fill it later in the original tube. Okay, so I think you all are done with it. Moving on to the next section. Okay, so now the next question is, so this is done. Name 3 layers of the human retina from outside to inside. Okay, now we are going to the eye. Okay, so name 3 layers of the human retina from outside to inside. Okay, so the first thing what you should do is look at a cross section of an eye. Okay, first let me tell you. Okay, name 3 layers of the human retina from outside to inside. The retina. Look at this diagram first. Okay, so you can see this diagram here. A very basic diagram of the eye. I mean, this is more than enough. You don't have to complicate things. Okay, the outside, wait. Okay, so the outside part is called the sclera. Inside is called the choroid. And then the innermost, innermost layer is called the retina. This layer is called the retina. So retina has 3 layers. Okay, so what are the 3 layers of the retina? Okay, so it has the outer pigmented epithelium. Again, this is directly from the resource book. Outer pigmented epithelium, number 1. Number 2, middle photoreceptive layer, and inner layer with neurons. So inner layer with neurons. So these are the 3 layers of the retina. This retina, if I zoom this part, okay, it will have these 3 layers. You will see outer pigmented. Outer means outside. Inner means inside. This white color zone, that is the inner one. So inner layer with neurons. So these are the 3 things. Okay. So this is direct theory. Next we will go to the question. Then we can discuss more. Okay. Briefly indicate. So this is the part. Okay. Look at that. And the next question is briefly indicate the distribution of the sensory cells in the human retina. Okay. Let's see what these sensory cells are. So again, we will come back to the diagram. So I will erase this. You can take a screenshot if you want, but this is not that important. These are the 3 layers. Okay. So I will erase this. Okay. So when they talk about the sensory, so they're asking us to indicate the distribution of the sensory cells in the retina. First, we should remember there were 3 zones, right? So in that, the sensory cells are located in the middle zone. The middle photoreceptive zone. Photoreceptive. So again, always use the names. Photo means what? Light. Okay. Receptive means almost like sensitive or sensitive. Okay. So these are the sensory cells. So what are these light sensitive cells? These light sensitive cells are the rods and cones. These are the two light sensitive cells. Rods means what? Cones means what? So what's the function of rods and cones? Again, use the word itself. Cone starts with letter C, no? So that means it helps in color. Then rod is just black and white vision. Okay, so this is how you remember it. So light sensitive cells. So then how does this light sensitive cells distribute? That is also another interesting part. So let us again look. These light sensitive cells, they are mainly present where exactly if you go back and see. So look at this. This is how a light ray will travel now. This at this region. Okay. So what do you say in the center? So that means at the posterior. So that means at the posterior part means back. At the posterior part of the retina, at the center of the posterior part of the retina, this region is called the yellow spot. Yellow spot. Okay. So you call this a yellow spot called macula lutea. That's all from the resource book. So what you all should do is go and read the resource book in these sections, but you understand the topic here. So at the posterior part of the retina, the center of the posterior part. So posterior means back, no? This is the posterior direction, the back direction. Okay, so posterior. And then center of the posterior part is called yellow spot called macula lutea. And this yellow spot, exactly at the center of the yellow spot, can you see there's a small depression here? This depression, wait, yeah, can you see there's a small depression here? Can you see? So I will erase it and show you. See, there's a small depression here. This is called the fovea. Okay, fovea centralis. And in the fovea, in this fovea only, but you only have cones. So this fovea has only cones. Cones means only for light, only for color reception. So this has only cones. Then what should you say? Towards the anterior part of the retina. So when you go towards forwards, both ways forwards, when you go, the number of cones decrease. That means there are fewer cones. There are fewer cones. When you go forward, there are fewer cones than rods. That means rods are higher, cones are low. But here, the center part, in the middle of at the center of the yellow spot, that is the fovea, you have only cones. So you can see cones only, and when you go forward, cones decrease and rods increase. Okay, so that's what you have to say there. You can take a screenshot here and keep this. So this explains everything. If you have any doubts, now you can put it. Part A is over. You can send any doubts. Feel free to send anything. Also, if you didn't get the PDF, send a WhatsApp message to that number. If you all understood, just say understood so I can move on to the next section. Okay, so I'm going in the next section. Then the next question is, okay, so now we are again going to osteology, okay? Bones, skeletal system. So how many bones are there in the cranium of the human skull? Again, going back. When we look at the skull consists of what? It consists of the face bones, and the cranium. Okay, cranium. So cranium means, I mean, the one that covers the brain mostly. Okay, the one that covers the brain. Okay, so how many bones are there in the skull? Totally 21 bones. Then the cranium consists of 8 bones. Face consists of 13 bones. 13 bones, but I will show you the difference. So then this has, there's a share. That means both the face and cranium has one bone in common, which is the frontal bone. Frontal bone. Okay, so now what I want you all to know is, it's very hard to remember everything now. So you have to have a method to remember everything. So in the face and the cranium, just I will go through this fast. These 8 bones and 13 bones, what's the way to remember? You remember a mnemonic. So I remember this. Pest of 6. Pest means what? Pest is like, I mean, if you hate something, a pest is something that destroys you or something. So now very hard to remember all these skull bones and cranium bones. So pesto, this is a pest of 6 type of bones. So that's how you remember. Okay. So what is this P? So now you know, no? P means what is this bone? So E, ethmoid bone. Ethmoid bone. P, eh, P is parietal bone. Parietal bone. Ethmoid bone. So you can see sphenoid. Sphenoid bone. No, yeah, sphenoid bone, and T, temporal bone. Temporal. O is occipital. And then finally F, frontal bone. So you can see all these 6 bones here, 6 type of bones, but then totally there are 8 now. So there, what are the paired bones here? It is the parietal bone and the temporal bone. So these are the paired bones. Okay, all others are single. Then you can count and see totally 8. Again, in the face also, this is one of the mnemonics I used to remember. My mandible, mandible means jaw, no? My mandible choose 9 very large uh, zucchini pizzas. So this is what I remember. You all go and search what is zucchini, okay? It is a type of vegetable. So here you can see M means maxilla, okay? This is maxilla. Mandible, it's there in the answer, in the word itself. C means what? Cheekbones. Oh, wait, I'll show correctly. So you say conche, no? Conche bones. And then here N is for nasal bones. This is for warmer. This is for lacrimal bones. Z is for zygomatic bones, zygomatic, and then P is for palatine bones. Okay, so in this also you can see like this, you remember all the types of bones. So here you have to remember there are only all the bones are paired bones except 2 bones. What are the 2 bones? The mandible, that is 1, and vomer is also 1. Okay, now I think I wrote it clearly. I hope it's clear enough. So then just take a screenshot of this and then we'll go on to the next thing. So here, my mandible choose 9 very large zucchini pizzas. That's how you remember all the face bones. Cranium is it's very hard to remember. No? So pest of 6. Okay. P-E-S-T. Pest of 6. Okay. I hope you took a screenshot. Next. So here now the question here is how many bones are there in the cranium of a human skull? I showed you 8 bones. Okay. 6 types, but 8 bones. So the answer is 8. In the cranium, name the types of pair of bones. That means what are the paired bones? I told you there are two paired bones. What are the 2 paired bones? The parietal. So P, parietal, and temporal bones. So these are the paired bones. So in which bone contains the foramen magnum? Foramen magnum means, so in your skull, if you have your skull, you can clearly say I'm a good artist. Okay, this is the thing, and this is the vertebral column. So in the skull, at this base, you have a hole or opening for the spinal cord. From the brain, a spinal cord should go, no? For that to go in the vertebral column. So that opening. So what do you call? So when they ask this question, I mean, it's not properly mentioned, but the answer for this is the inferior surface of the cranium. So basically they're asking, where is the foramen magnum? So you say inferior surface of the cranium. Okay, cranium is skull bone. Okay, in which bone is the inner ear located? Inner ear is located in the, all these are theory, so nothing much to remember. I mean, as understand, but a lot to remember. Temporal bone. Inner ear is located in the temporal bone. Then what's the next question? What are the fused bones in the human vertebrae? Fused bones come at the end. Okay, so vertebrae consists of what types of bones? Again, I will, yeah, go back there. Vertebrae consists of what types of bones? So when you see from here, skull, and then now vertebrae. So this is a very simple diagram. There is cervical vertebrae, thoracic vertebrae, and lumbar vertebrae. And then finally the fused bones, which is the sacrum and the coccyx. This has 5 fused bones, this has 4 fused bones, and both are triangle shaped bones. Okay. So yes, you can take a screenshot of this. Okay, so you take a screenshot of this and ask any doubts if you have. So and if you understood, you can put a confirmation in the chat. I hope you took a screenshot of this. Okay, and now Okay, here. And you can even take a screenshot of this if you want. There is okay, so take a screenshot of this. Are we supposed to memorize the whole resource book? A good question. Thing is, obviously no. Even when I went for the exam, there are several chapters even which I didn't even have a look at. So what you should do is, no one can memorize the whole resource book. It's not possible, no? So have methods to memorize the important stuff. So now you know human form and function, plant form and function, chapter 2. All those are very important chapters, no? So spend most of your time to do that, to study that. And understand that the rest of the chapters which I mean, you know, when you look at the past papers, you know what is not important. So those kind of chapters have a idea of it, but not memorize, not necessarily to memorize. And try your best to use methods to understand more than memorizing. Okay, you can use mnemonics and such things, then it'll be easier. But at the end of the day, you have to do hard work now to achieve it. So that is it. Okay. Next, draw and label the first typical cervical vertebra. See, skeletal system is important. Draw and label the first first typical cervical vertebra in human vertebra. Yes, first typical cervical vertebra in human vertebra. So again, there is nothing for me to do other than show you the answer itself. It's there directly in the resource book itself. So here you can see, so these are the, so first what you should know is cervical vertebra means exactly under the skull. So this region, cervical, the neck, cervical means neck. So then it should have the smallest. So it is the smallest vertebra and the body is also very small. Body means what? This always the front part is the body. So every vertebra has this common thing. The front is body. Wait, I'll quickly tell you. Front is body, and then so you will have the bones here, and you will have like this transverse processes, and then back you will have a vertebral canal for the vertebra to go, and then you will have a spinous process. Normally, it should be something like this. So remember this. So the body, so front is body, and then transverse processes here. So this is directly from your resource book. Body, transverse processes, and then in the transverse processes, you have foramen. So this is a special thing. Foramen for vertebral artery, and then vertebral foramen you can see here for this spinal cord to go. So you can see this is big, bigger than usual. So then remember the differences. Then it's easy. Okay, and then bigger than usual. So you will check bigger than usual means compared to other vertebra, lumbar, transverse, thoracic vertebra, and then bifid spine. Okay, bifid. Usually a spine is like single, but here bifid spine, and then articular. So these are the things they expect when they ask about what is the first typical cervical vertebra. So just take a photo, I mean a screenshot of this and keep. It's there in your resource book also. Okay, and I'll move on to the next one. Okay, so that's also over with. Next question. Again, now we are coming to skeletal muscle. So here also name ABCD mentioned in the above diagram. So this is, first of all, when you look at this, what should come into your mind? This is a sarcomere. Sarcomere means what? Simply, it's a skeletal muscle cell. Sarcomere, skeletal muscle cell. So that's the idea here. Not exactly skeletal muscle cell, but a part of skeletal muscle. So okay, yeah, so forget about that. Okay, so sarcomere. So what are the parts of sarcomere? Again, that also you all know. I'll show you another picture. Yeah, so remember this, okay? So this is a sarcomere, okay? Basically, can you see the screen? Yeah, I hope y'all can see the screen. Okay, so this is a sarcomere, okay? So sarcomere means it consists of, what is the definition of sarcomere? Again, I'll go back to the question, okay? Sarcomere is from one dark, I mean, you can see these lines, right? From one line to another line is sarcomere. So then A is sarcomere. So what is this line which I showed you? That is B. B is the Z line. Okay. Then again, what is C? So C, you can see these are like, even though it's dark in the PDF, very thin. Okay. These are very thin structures. Can you see these thin? Okay. So you call them what? Thin. Thin means actin. Actin filaments. These are thin. And then finally, when you go to and look at D, D are the yes, so if you see here, here, can you see here? D are thick filaments. Okay, so thick means myosin. Myosin filaments. These are thick. Okay. Yeah. So that is basically it about skeletal muscle fiber. You all just go and check. Okay. So there are several things in skeletal muscle fiber. But just know this is sufficient, I guess. And then the next is we'll come back to it. Okay. So this is the first part. And then the next thing is, how does region A and B vary during contraction of a muscle? Contraction means what? Contraction means muscle should shorten. You know, no, that's the definition of contraction. So contraction shorten means what? A. What is A? This. Okay. So you can see here what happens to A. A will, oh, A means this length. Yeah, A will shorten. So again, B will come close to each other. Same meaning. Okay, come close to each other. So then B come in close, A will shorten, B come close to each other. That means basically the Z lines are coming to each other. That only we are expecting you to know. Z lines are coming close. Okay, done now. Next, what is the function of calcium ions in muscle contraction? So when we come to sarcomere, I will just give you a small recap. Sarcomere, what should you know in skeletal muscles? You should know something called the slide in. Wait a bit. I think, can you all see my screen now? You all can see my screen, right? Yes, I think you all can see my screen. Next. So when we are talking about sarcomere, you should know about something called as side-in filament theory. So this is the most important thing you should know when it comes to skeletal muscles. Okay, so in slide-in filament theory, here, so I will just show you this diagram. Can you all see the diagram? Can someone soon say whether you all can see the screen? Can someone say, can someone tell whether you all can see the screen clearly? Wait. Okay. Wait a bit. Wait. Okay, now it's okay, it seems. Okay, so now you can look this diagram. I'm trying to show you a quick diagram. Sarcomere means what? Sarcomere means from one Z line. So what is this Z line? Okay, sarcomere is from one Z line. Here I'm drawing a Z line. Here is a Z line. This line is called a Z line to another Z line. Okay, now so that's only sarcomere. So when we are talking about slide-in filament theory, this is the theory that they made up to explain how a skeletal muscle contracts. So how does that happen? This myosin head, this is called the myosin. Okay, I told you the middle ones, the thick ones are myosin. It has a head here. You can see circles like this head. That attaches to this actin. Actin is thin. You can see thin filaments. So attaches to this actin. You can read, I'm telling you the basic idea, and then it pulls it. It attaches to the actin and pulls it this direction. So this direction, can you see? So when it pulls it towards each other, Z lines come towards each other and the muscle contracts. So that's the basic idea. Okay. So what you should know here is for this to happen, okay, for this to happen, this, you can clearly see here, this muscle, myosin head should bind to actin, right? Normally, it won't bind to actin. Okay, normally for anything to happen, there should be an activation. Or else every time your muscle will just be contracting. No. So for that, what happens is there is something called a sarco. So endoplasmic reticulum in the skeletal muscle is called what? Skeletal muscle is called sarcoplasm. Everything with sarco means muscle. Sarcoplasm. From this sarcoplasm, calcium ions are released. These calcium ions will come to this actin. Okay. Actin means what? These are thin filaments, right? It consists of both. Actin consists of both. Actin means, so this thin filaments has two things. Okay. It has something called a tropomyosin. It's not clear, no? Right. It has something called a tropomyosin filament and something called a troponin. So both are proteins. All these are proteins now in muscles. So this circle-like things are like troponin, okay? And other one is tropomyosin. So what happens here is this myosin wants to bind to what? The myosin has to bind to these actin filaments, no? So to bind, it has myosin head binding sites, myosin head binding sites in this actin. But that is being covered by this troponin. So what happens? This calcium which are released from the sarcoplasm will come and bind to these troponin molecules, and expose these binding sites. Then only this myosin can bind to this actin and then pull it okay, towards the center. So that is only this idea. So what is the function of calcium if they ask? It is to expose the myosin head binding sites in the actin filament. That's the answer. Okay, now? That is it. If you want, you can take a screenshot of this messy diagram. Up to you all. Okay. Yeah, okay, I'm changing. Okay, then finally explain the nerve supply for skeletal muscle fiber. Nerve supply for skeletal muscle fiber. You know skeletal muscles are under your voluntary control. It's under your control, right? So everything which is voluntary control is somatic nervous system. If it is not in your voluntary control, autonomic nervous system. Okay, that's what we want you to know. Yeah, if you want, you can take a screenshot of this, and my section is over. You can take a screenshot of this. If you all have any doubts, this is your time to put it. What is the use of calcium ions? There is a question. What is the use of calcium ions? I hope you all understood. But remember what I told you. Calcium ions go and bind to troponin. Take a screenshot of this. Fast, I'll go there. Where did I write it? Here. So you remember here. If you see this diagram, I think give me one moment. Can you see this diagram here? Yeah. So calcium ions, what do they do? Wait. So calcium ions go and bind to troponin. Is it clear? Yeah. Okay. Here, calcium ions that is released from the sarcoplasm goes and binds to troponin. Okay. Troponin is also a protein that is present in actin. All this actin is this thin filaments. So when it goes and binds to this troponin, then it will make this troponin move somewhere else. That will expose the actin, that will expose the myosin head binding sites in actin. Yeah, so that is the way you answer the question. So what is the function of calcium? Calcium binds to troponin, okay, binds to troponin and exposes the myosin head binding sites on actin. So that is the answer. I hope you all understood. If you all have any other questions, you can ask now. And that's it. Okay. So, okay, thank you.
Okay, so we'll continue with the structure paper. So next we are going to see the fourth structure. So fourth structure has three components, A, B, and C. A is about unit genetics and the molecular biology. B is from environmental and C is from the microbiology. So I'm not going to do microbiology because it is directly from the resource book. So I'm not going to explain. You can just look at the answers or else go through your resource books. We will be focusing on the parts A and B today. So if you have any doubts, you can unmute and ask. Please don't put in the chat because I won't be able to see your doubts. So if you want, please unmute and ask your doubt anytime you want. Okay, let's start. So the first question is about the non-Mendelian inheritance. So the definition is asked. So usually in structure type questions, there is a tendency to ask the definitions of the very main terms that is in the biology. So one of that is non-Mendelian inheritance. There is Mendelian inheritance as well as non-Mendelian inheritance. So non-Mendelian inheritance. So now there are Mendelian laws, right? So these non-Mendelian inheritance won't follow them. So they will not segregate according to the Mendel's law of inheritance. They will have rather different things. Okay, so the phenotypes that they won't appear in the ratios that is predicted by the Mendelian genetics. Okay, so that is what non-Mendelian inheritance is about. So if they ask you what is meant by non-Mendelian inheritance, so you can say it is the inheritance patterns in which traits do not segregate in accordance with Mendel's law of inheritance. So what I told now was directly from your resource book. From the resource book itself, I've told the definition. So they want by the rules of the Mendel's law of inheritance. They do not go with that. They will segregate in a different manner. So there are many examples for this non-Mendelian inheritance, right? So can you tell, can any of you tell what are the examples for non-Mendelian patterns, you know? Fine. So if I wait, the time will be going. So I'll just say, so the examples for non-Mendelian patterns, you know, there are incomplete dominance as well as co-dominance. There is also complete dominance that we will see. Now, I was telling about the examples for non-Mendelian patterns. Okay, so you should be able to know all of them. First, since the next question is about incomplete dominance, we will see a little bit about the incomplete dominance. So first of all, the question is what is incomplete dominance, right? So incomplete dominance, if there is incomplete dominance, there should be complete dominance, right? So incomplete dominance, it is like a phenomenon where the dominant allele, so it's quite easy, where the dominant allele will completely mask the recessive phenotype. So it will completely mask the recessive phenotype, where it will result in similar phenotypes for both homozygous dominant zygote as well as heterozygous zygote. So if it is homozygous dominant as well as heterozygous zygote, both will show the same phenotype, where incomplete dominance, because the dominant allele will mask the effect of the recessive allele. But in incomplete dominant, it is not like that. In the heterozygous state, if it is a heterozygous state, the phenomenon of expressing the blend phenotypes from both alleles will be there. So the dominant and recessive both will contribute, and there will be a blend phenotype. Okay, so that is what we call as incomplete dominance. So the phenomenon of expressing blend phenotypes from both alleles is called incomplete dominance. There is no masking by the dominant allele here. That is why we call it as incomplete dominance. Incomplete dominance, there is some masking effect of the dominant allele, but here it isn't like that. So it is what we call as incomplete dominance. So there is a very famous example for incomplete dominance, right? So that is the Mirabilis jalapa, or the 4 o'clock plant. So there are several types of flower colors in this plant. Okay, now if next question is about that, let's see. In Mirabilis jalapa, there are several types of flower colors. When red colored plants are crossed with white colored plants, all F1 hybrids have pink flowers. Explain the occurrence of pink colored flowers using a genetic diagram. Now you have to draw a genetic diagram. So if it is complete dominance, you either get white color flower or red color flower. But here you are getting a blend. So if you add when you add red color paint and white color paint, you get pink color paint. Likewise here, the both of the alleles contribute and they give a blend phenotype. So let's see how it was done. So we can write it like this. First, we should know the P generation. Okay, the red flower crossed with white flower. So we can write the genotype for red flower, CR. So this is the traditional way of writing this. Okay, so here's CW and CW. So next, you should write the game. So this is the order of writing. If you miss the order, you might not get marks. If you miss some line in between, you will lose marks. So be careful. careful in writing. So here gametes, it's completely CR. So you can write CR, which is 100%, right? 100%. Here again, CW, it is 100%. Don't forget to circle it. Now, you will get the F1 generation. What will you get? CR and CW. Now, there is a mix of both LL. So, what is the color you will get? So, here you will get pink color. Pink color flower. Again, for F2 generation, you can cross them both. Okay, so you can use the square, right? So CR, CW, and CR, CW. So for F2 generation. You can do the other way also, but this is easier. So write down sperm and egg here. This is also in your resource book, I guess. So you can refer it to that too.
Well,
so you just have to do it like a multiplication. CR, 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 CW, CR, CW, and again CW, CW. So what is the genotypic ratio? So the genotypic ratio is CR, CR is to CR, CW is to CW is to CW. It is 1:2:2:2, 2:1. So you can count it. So the phenotypic ratio is also similar to the genotypic ratio. Okay. So here you have red color, pink color, and white color flowers. So 1:2:1. So this is the way you have to write the answer for the question B question of the second question. So this is the way you have to answer. This is also in your resource book, I guess. Please check it. So this is the way you have to answer there. So that is all about the incomplete dominance, and we have seen an example. So now we have done incomplete dominance. Please try to go through complete co-dominance, polyallelism, polygenic inheritance, gene linkage, everything. Try to go through. So now the thing is, for essay, of course, you can skip these units, because if you are completely thorough with 2, 3, 4, and 5th units, 2, 4, and 5th units, you can write more than 3 essays, of course. But then for structured questions, you will definitely get unit 6, 7, 8, 9, as well as 10. Now they ask more questions on unit 10 too. So don't skip any units. At least try to remember everything for the structure. If not for the essay, at least try to remember for the structure, because mostly they are like one short sentence or 2 short sentences. So try to remember. Now the next question, so 3rd, 4th, and 5th, they are from molecular biology. So molecular biology has 2 parts, right? Molecular biology and recombinant technology. So here, let's see. For first question is, what is meant by protein trafficking? So what is meant by protein trafficking? So there are certain polypeptides. Okay, so there are certain polypeptides. They have some additional segments, but than what is required for its function. So those segments are not necessary for their function. It's not necessary for their function, but it is there. Because they act as signal peptides. They will act as signal peptides, and they will guide the polypeptide to a particular location in the cell. So, like for example, Golgi apparatus, or to be secreted for exocytosis. So it will guide. So it's like a signal peptide. It guides the polypeptide to a particular location. So this is what we call it as protein trafficking. So protein trafficking is, there are certain polypeptides which have additional segments than what is required for their function, but they act as signal peptides and guide the polypeptide to a particular location in the cell. Or to be secreted. So this is what we call as protein trafficking. It is quite easy and from the resource book. Okay. So next question. Name the most commonly used DNA joining enzyme. So there are DNA joining enzyme, right? So they are asking for the most commonly used DNA joining enzyme, which is used in the gene technology. And they are asking what is the source of this enzyme. So when you are studying molecular biology, you might have studied different types of enzyme, which is useful for DNA replication and all the other process in molecular biology. Okay, so there is a most commonly used DNA joining enzyme, which is used in gene technology. So what is it? It is the T4 DNA ligase or ligase, whatever you call. T4 DNA ligase. Okay, now this ligase, what their function is to, of course, DNA joining enzyme. What they do is they will join the DNA fragments from different sources. So we will cut DNA from different sources and we have to join them. Okay, so we have to join them by forming a phosphodiester bond. For that, this DNA joining enzyme, which is DNA ligase, is used so that it will be joined together by the phosphodiester bond and obtain a recombinant DNA molecule. So for a recombinant DNA molecule to form, we will get DNA fragments from different sources. And then we will use this enzyme to form phosphodiester bond between them and join them completely. Okay, so this is about the DNA ligase. We call it as T4 DNA ligase. Why? Because the source, they asked the source also. The source is from T4 bacteriophage. T4 bacteriophage. So since we get it from T4 bacteriophage, the DNA ligase is also called as T4 DNA ligase. Okay, so that is about the DNA ligase. Okay, now let's move to the fifth question. So if we see the fifth question, it is about PCR. So you all are quite familiar with the PCR because it was a very common word, common thing we use. So this is about the PCR. Sometimes it is also a question that can be asked in essay, because you can see it was in 2020 or something. They asked in an essay for PCR. So here there are these two other very simple questions from PCR. First is, what are the ingredients of a PCR mix? So what are the ingredients of the PCR mix? So you will need, first, what is PCR? So PCR is like you are copying a sequence of DNA in vitro, like the DNA replication. So you do the DNA replication outside your body. You are not doing inside the body. You are doing outside the body in vitro, that you are doing a DNA replication where you copy a particular DNA sequence. That is PCR, polymerase chain reaction. Okay, so there are some ingredients that we should use for this PCR. So you will need deoxyribonucleotide triphosphates, especially DATP, DGTP, DTTP, DCTP. Okay, it's better if you write all of them, but it is fine if you just write deoxyribonucleotide triphosphate. And then you will need a single stranded template DNA. Okay, single stranded DNTPs, single stranded template DNA. And then you know that you need a DNA primer, otherwise nucleotides cannot be joined. Okay, you need a DNA primer so that the nucleotides will be joined further. So DNA primer is, it has like a small number of nucleotides, so that therefore you can call it as oligonucleotide too. Oligo means few molecules, right? Few number. Then you need magnesium 2+ ions. Then Tec DNA polymerase. So where do you get Tec DNA polymerase from? It is from a thermophilic bacteria, right? Thermophilic bacteria called Thermus aquaticus. They might ask you in MCQ. Why don't we use just DNA polymerase? Why do we go for Tec DNA polymerase? Why do we use stacked DNA polymerase? Why can't we just use DNA polymerase? It is because at some point we will heat it for 95 degrees Celsius, like very high temperature. I think it is 95 degrees Celsius, where the denaturation is done. Okay, it is by heating the PCR mix to 95 degrees Celsius. But at this temperature, you know, right? So enzymes have a optimum temperature. So for example, if we take our body enzymes have an optimum temperature which is equivalent to our body temperature. So it will have the maximum activity. But when it is increased, when the temperature either increase or decrease, that activity will be reduced, right? Just like that, now 95 degrees is like very high. It is close to 100 degrees Celsius. So if it is 95 degrees Celsius, then only there will be denaturation. So most of the enzyme will also denature. So therefore, we should, it is necessary that we add some DNA polymerase after the denaturation. But since we have this thermophilic bacteria where we can get this stacked DNA polymerase, we use it as we mix the PCR mix. Okay, so that is the reason why we use stacked DNA polymerase, because if we just put the DNA polymerase, it will denature at 95 degrees Celsius. So I didn't explain the process of PCR, so I hope you know. Yeah, next, give to, that is all for the ingredients of a PCR mix. Now, if you read your resource book, all of these are not in one area, right? So you have to collect it from each of the sentences. So we have made it easy for you, and these are the ingredients of the PCR mix. Now they're asking for 2 applications of PCR in medicine. There are many applications really. So there you can analyze the clinical specimens for the presence of infectious agents like HIV, hepatitis, malaria and all. Then you can analyze the mutations causing genetic diseases, for example, cystic fibrosis, sickle cell anemia, phenylketonuria. Not only this, also used in forensic laboratories, and PCR can make a large number of copies from a small number of template DNA. So it is useful because only a tiny amount of original DNA is required. Okay. Then PCR is also an essential technique in cloning procedure. Likewise, you have so much of applications straight from your resource book. So please refer to your resource book. I think 8 or something is there. So just refer to it, and you can try to remember at least 3. Okay. If they ask in essay, you might have to write everything, but then for a structure, at least try to remember 3. So this is all about A part. Now let's move to the B part. So B part is also quite direct from your resource book, which is from environmental biology. I know environmental biology is a very boring chapter because you have to remember so many stuff there, but still you will have a structure question, like a part of a structure question, so it is better to know. Let's see. First question: distribution of major vegetation types in Sri Lanka is based on 3 major criteria mentioned there. So this is also in your resource book, right? So this comes under ecosystems of Sri Lanka. So you know Sri Lanka is in the North Equatorial Tropical Zone. So therefore it will experience a climate with high rainfall and temperature. Due to this, there is a wide array of terrestrial and aquatic ecosystem, right? Because of these changes, we have a good wide array of terrestrial and aquatic ecosystem. So we can divide the ecosystems like into 3. I hope you know it is terrestrial ecosystem, inland wetland ecosystem, and ecosystem with coastal areas. Okay, so these are the main broad types of the ecosystem in Sri Lanka. Now they ask what are the major criteria of the distribution of the major vegetation types. So you know that is from your book. It is mainly the local climate, mainly the rainfall and temperature, then topography and the soil conditions. So these three determine the distribution of major vegetation types in Sri Lanka. Okay. Next, some special features of the terrestrial ecosystem of Sri Lanka are given below. Name an ecosystem for the special feature. So if you should answer for this question, you should know all the features of each terrestrial ecosystem, right? At least have a broad idea of the ecosystem so that you will be able to identify. Let's see, twisted stems. So where are twisted stems present? Twisted stems are in the tropical montane forest. So I'm not going further deep inside because these are from your resource book. Please try to read and understand. So twisted stems are there in the tropical montane forest. Epiphytes in branches are found in the tropical wetland, lowland, rainforest. And third one, thick grass cover with few scattered trees. Okay, thick grass cover with few scattered trees. And then, so these are the answers for the second question. Third one, what are the characteristic features of a mangrove ecosystem? Okay, now there is mangrove ecosystem. You should know the main character. It is better to know the mangrove ecosystem for the essay. Sometimes they might ask you a short note on mangrove ecosystem because it's an important ecosystem in Sri Lanka. So you know mangrove ecosystem is like an intertidal vegetation that covers the fringes of the lagoons and estuaries. So there are lagoons and estuaries. There will be an vegetation. The vegetation that covers these areas are the mangrove ecosystem. So they will share characteristic usually like saline or brackish water because they are close to the ocean. Brackish water. They will have loose soil and anoxic condition. What is anoxic condition? It means there is lack of oxygen, anoxic condition. And also the vegetation will be exposed to intense sunlight. Wait a bit. Right, so these are the features of a mangrove ecosystem. Okay, these are the features of the mangrove ecosystem. I hope you know there are mangrove plants which can be categorized into two groups, which are the true mangroves, right? So you should know that also. Now, next, give 3 adaptation of mangrove vegetation with their functions. So these mangrove vegetations have some adaptations specialized for certain functions. We'll see. They will have leaves with thick cuticle. Okay, this is to cut off the excess radiation or to reflect the sunlight. Okay, so they will have thick cuticle. So you know this already because you might have studied in xerophytes also. So the thick cuticle will cut off the excess radiation. It will reflect the sunlight. And there are salt glands. First one, leaves with thick cuticle. Thick cuticle to cut off radiation. Then salt glands below the surface. There will be salt glands below the surface. It is to secrete the excess salts that are absorbed. So there will be salt glands to secrete the excess salts that are absorbed. And there are pneumatophores. which will help to intake the atmospheric oxygen. It will help to intake atmospheric oxygen, and finally viviparity. It will help to establish seeds successfully soon after falling from the tree. Okay, so these are the adaptations from the, so there are 4 adaptations I told, so you can write any 3 of them. So these are the adaptations and their functions, right? So these are the functions. So these all are directly from the resources. There's nothing to explain, you just have to remember. Next, mention 4 factors that contribute for desertification. Desertification means loss of the vegetations. So main climatic variations, over exploitation of water and soil. If you use too much, then it will lead to desertification. Then excessive use of agrochemicals. Use of agrochemicals. Also, poor land management practices. Poor land management practices. Okay, so these are the factors that will contribute for the desertification. Finally, name a Ramsar site located in southeast of Sri Lanka. So what is it? Just directly from the resource book, Kumana Wetland Cluster. Okay, that is all for the structures. Structure part today.
Okay, I hope you all can hear me. So we'll again just go back. Now we'll just look at some MCQs. Okay, so again, animal form and function MCQs. Let's start with question number 26. Okay, so here again, respiration. Again, you all can look at the screen. Question number 26. So here, when they ask about respiratory structures, this one is basically direct from the theory. You know, there is nothing to understand here. So what you should know here is when they speak of body surface, which type of animals have which type of animals use the body surface itself as respiration. These are the animals that are very small. No, small in the sense they are very tiny, that their surface area to volume ratio is very high. So then they can use directly what diffusion through the body surface. Diffusion through the body surface is more than enough for them. So what are those? Then it's the primitive ones. No, so Cnidarians, then flatworms, okay, so platyhelminthes, and then earthworms also are special, earthworms. So these are annelid, no? Earthworms are annelida. Likewise, gills. So gills means then you know there are 2 types of gills, external and internal gills. Okay, so external gills who uses marine animals and annelids, so marine annelids, okay. And then internal gills, so now this is directly from theory, so nothing like nothing much to understand here. So fish, shrimps, prawns. So yeah, so that is it. And then when you look at lungs means so obviously mammals, humans like us, mammals, reptiles, okay, and birds. So you know the mammals and birds are close to each other. Okay, so mammals and birds. So then they have lungs, and then reptiles also. Then again, tracheal system means insects. Okay, and skin means who has the most sensitive skin when it comes to anything. So even when an environmental change or disaster is going to happen, who detects it first? Amphibians. If you see the toads and all, they detect it very fast and they will move away from the disaster. That is because they have very sensitive skin. So then again, amphibians. Okay, so they use their skin. Amphibians. They have very what do you call, wet skin. No? So now that is the basic idea of the respiratory structures. Then they ask in which respiratory structures do phylum Arthropoda have. Arthropoda means insects, those kind of stuff. So then what's the answer here? Gills. So when you look at gills, Yeah, so arthropoda means, no, so they have several legs. So then again here gills means internal gills. So gills are there. And then next, and then tracheal, no, not tracheal system, book lungs. So book lungs, so here when you look at book lungs, I think I missed it, no? Book lungs, so then spiders, so then C, and then again E, tracheal system, insects. So B, C is the answer. Okay, I hope you all got that. You all can take a screenshot if you all need to, but that is directly theory. Okay, so B, C, E. Even reptiles, yeah, okay. B, C, E. Even D is right, but B, C, E is the answer. Okay, then moving on to the next question. So question number 27. So again here, when they ask about vibration receptors, again it's coming to, yeah, so then here it is the ear, no? So they are mostly talking about the ear here. So when we are talking about these vibration receptors, what do you call these vibration receptors? They are also called as mechanoreceptors. Okay, so mechanoreceptors are also, so now there are several types of receptors, right? Mechanic means what? Some kind of pressure or touch or some mechanical stimulus. Okay, so then there are three types of mechanical receptors. What are the mechanical receptors? Touch receptors. So touch means what? Small pressure, very little, less pressure receptors, just touch. Then other one there is pressure receptors, and then there is vibration receptors. Okay, so we should know that. So then from the touch receptors, you should remember. So the touch receptors usually start from letter, usually from the letter M. So what are the letter M things we remember here? So Meissner corpuscles, Merkel discs, and then free nerve endings also. So free nerve endings. So these are only the touch receptors. So what does Meissner corpuscles detect? So they detect the light pressure. They detect, so Merkel discs, they detect light touch. Okay, so this is light pressure. This is light touch. Yeah, I hope you all can see light. So that Meissner corpuscles detect light pressure, Merkel discs detect light touch, and free nerve endings. So that's all. That's about touch receptors. Okay, so letter N is the thing you have to remember. And moving on, when we talk about pressure receptors, again, so pressure means P, no? So remember P. P for what? Pacinian corpuscles. Okay, so P. P for Pacinian corpuscles. And then this detects what? Deep pressure. So what we started off by telling what touch means small pressures, no? Light pressure, light touch is touch receptors. Pressure means deep pressure. Deep pressure, Pacinian corpuscles. So it's easy, no? P and for T, touch receptors, remember letter M. And then we are going to vibration receptors. There. So all these are mostly, I mean, all the receptors, touch pressure receptors, they also come under vibration receptors. So they also can detect these vibrations. So then that includes Meissner corpuscles, and then Pacinian corpuscles, and then the hair cells, the special hair cells that are present, special hair cells present in the organ of Corti in the inner ear, and then hair cells in the vestibule of the inner ear. So all these are directly from the resource book itself. So when it comes to animal form and function, you should like know everything. No? Animal form and function is the most important thing. So hair cells of the vestibule of the inner ear, and then again, so then all these are hair cells, and then finally hair cells in the semicircular canal. Semicircular canal. So here, what you should remember here is, so now you can see here, all these are, so again, all these are hair cells, all are in the ear. So now what is the function? For us to remember the function, wait, so here the hair cells in the organ of Corti, what do? They detect sound. Okay, so they detect the sound vibrations. All these are vibrations. So they detect sound vibrations. The hair cells in the vestibule detect gravity. Okay, and the hair cells in the semicircular canal detect motion. So now this is the basic idea of the entire mechanoreceptors. So now what is incorrect about the mechanoreceptors? Sound. So let's read from number 1. sound vibrations are detected by organ of Corti in the inner ear. So you can see here, no? Organ of Corti is here. So they detect sound vibrations. Yes, so that is right, true. But they are asking what is incorrect. So we have to find what is false. Intensity of sound is detected by semicircular canal. So you can come here, hair vestibule of the inner, no, hair in the semicircular canal, motion. So here, intensity of sound, no. Not the loudness, no, not the intensity of the sound. So then this is the false answer. Other one, Meissner corpus and Pacinian corpus is a vibration receptors, true. Vestibule of the inner ear contribute to maintain balance. So again here, when they say gravity and also position, no? So you should know your position. So yes, it will help to maintain your balance. If you know your position, you are in balance. Okay. And then finally, most touch receptors. Yeah, touch receptors are also detect vibration too. Can you clearly see here now? Touch receptors, Meissner detect vibration too. So the answer is 2. What is incorrect? Intensity of sound. Yeah. I think you all understood. If you all have doubts, you can put, I mean, in the chat. Okay. Moving on to the 28th question. Yeah. Yeah, you can hear me now. Okay, so moving on to the 28th question. Which of the following is a function of the liver in addition to its role in digestion? So first of all, should I? Okay, I will erase this. You can take a screenshot of this. I'm going to erase it. If you all have doubts, you can ask. Okay. Take a screenshot of it. Okay, finally, 28th question. We are looking at the 28th question. Which of the function is which of the following is a function of the liver? Okay, so first of all, we should know liver. Is it a part of the? So when you remember the gastrointestinal tract, okay, from the stomach, don't judge the diagrams. It's basically something like this, J shape, from the stomach, and then you will have the intestines like this, the small intestine, and then you will have the large intestines. So the diagrams are a little bad, and then you'll have the large intestine like this, and then it'll come to the rectum, and that's it. Okay, so then here what you should know here is kidney is something not at all attached. It's somewhere in the right side of the body. It's not even attached to this digestive system that is going on here, the intestines. It's not even in the digestion or the absorption directly. It is not doing anything directly. So that's what you should know. It's something, the kidney is something here that would put, I mean, it would send structure. So what does the kidney release? Kidney mainly is the main function of kidney when it comes to digestion of food is by producing bile. So it produces bile. It produces bile, stores it in the gallbladder, and the gallbladder puts the bile into this intestinal system. So then the function when it comes to digestion is its production of bile. It never directly comes and involves in the digestion. Same as absorption. It doesn't directly absorb. No. What is absorbed by the intestine into the blood only, it takes and stores. So its function is store it more than absorption. Then let's see what's the answer here. So first is we will start with number 1. Contributes for glucose homeostasis. When it comes to liver, liver's main function is what? Storing. I told here, right? Storing. So when take anything, it will store. Proteins, it can store. Proteins, it stores. Even though main protein stores are in muscles, yeah, proteins also it can store. Amino acid, it stores. Okay. So here, amino acid storage, it does. Say any storage. Glucose, yeah. Glucose storage, yes, it stores. And then fats also it can store. Likewise, vitamins, it stores. Iron, it stores. So several things. When you take vitamins, minerals, almost everything is stored in the liver. So then glucose homeostasis. What is the meaning of homeostasis? Homeostasis means maintaining a constant environment in the body. So when there's too much glucose in the blood, what happens? The liver takes up glucose. So liver takes up glucose and stores it. Likewise, when there is less glucose in the blood, then the liver releases glucose. Okay, so both of this happens under hormonal influence of what? When you are starving, you want to release glucose into the blood. So then you have to use the hormone glucagon. Likewise, when you have eaten a lot after a meal, you want to take glucose into the liver to decrease the glucose in the blood, then you use the hormone insulin. Okay, so to contribute to glucose homeostasis, yes, 100% true. Okay, then next, completes the reabsorption of water? No. I told it doesn't involve in any absorption directly. Reabsorption of water completion happens in large intestine. Okay. And who does the most reabsorption? Majority of reabsorption of water is small intestine. So that you should remember. So completes reabsorption of water? No. False. Okay. Synthesis of vitamins. So synthesis again? No. No. So synthesis it doesn't do. Store in vitamins. Okay. So synthesis of vitamins with help of microorganisms happens again in the large intestine and small intestine, as I remember. But again, it's the intestinal system. Okay. So again, that's also false. Then absorption of nutrients and recovery of water and ions? No. Again, absorption, everything done by the intestine. What does the liver do? Storage. Okay. So that's the false. Again, defense against microbes. So when coming back to that, when you know the liver's architecture, liver has some macrophages. No, special macrophages called Kupffer cells. Yeah, Kupffer cells. So special macrophages. So if you remember, they line the sinusoids. What are sinusoids? Those are like incomplete blood vessels within the liver. So you go and learn the liver. I mean, that's very important, liver architecture. So they have special cells called Kupffer cells. That's the phagocytic activity. They are phagocytes. So phagocytic activity means what? They destroy bacteria, right? So destruction of bacteria means they are defense. They are a defense mechanism against microbes. So this is also true. So which of the following is a function? So then they have us following function of liver. So then both 1 and 5 are true. So if they ask this in an exam, go with the most important one. The most important one is number 1. So the answer is number 1. So if you all have any questions, you can put it in the chat. That's it for my part. So if you have any questions, you can put it now. I hope you all understood.
Okay, so I'm also continuing with the MCQ. So I have selected a few MCQs from Unit 2. So we'll go through them. I think there are some mistakes, so we'll just try to correct them here. Yeah, so we'll start from the first question. So it is about polysaccharide. Okay, it's Unit 2. So polysaccharide, so you know carbohydrates, right? So I'm just going to quickly do this. So carbohydrates, you have 3 types. What are they? Monosaccharides, disaccharides, and polysaccharides. So these monosaccharides, disaccharides, you should know their properties where they're soluble, sweet taste and all, but polysaccharide is opposite to them. And also you should know that there are some examples for monosaccharides, some disaccharides, and also the polysaccharides. So now the question is about the polysaccharides. Okay, it is about the polysaccharide. Let's go through all the sentences, all the statements given. Okay. Right. Pectin is formed by galacturonic acid. So it is mostly about the monomers. So there are many types of polymers. So here we will be mainly focusing, we'll just go through a quick revision on the monomers. So if you see, there are some polysaccharides, you should know what are they: starch, glycogen, then cellulose, inulin, pectin, hemicellulose, and chitin. So you know what is the special about chitin? Chitin has nitrogen in it. Usually carbohydrates had carbon, hydrogen, and oxygen. The elemental composition of carbohydrates is carbon, hydrogen, oxygen. But in addition to that, in chitin you have nitrogen also. It is a special thing to mention. Okay, so these are the polysaccharides. You know, you should know the monomers of each of them. So the starch, glucose, and cellulose, all 3 has glucose. Glucose is their building unit or the monomer. Next, you have inulin. Inulin, it's fructose. So fructose is a monosaccharide, which is the most sweetest sugar, right? So it is the sweetest sugar. So it is inulin. Next, pectin. Pectin is galacturonic acid. Hemicellulose, it is pentose. Chitin is glucosamine. glucosamine. So you should know their functions also. Let's see quickly. The starch, you know, it is stored in plants. Glycogen, it is stored in animals and fungi. Cellulose is a component of the cell wall. So cell wall, even there is primary cell wall, secondary cell wall, right? In primary cell wall, you have cellulose. Then inulin, it is also storage of fructose. It is also storage of fructose in the tuberous of Dahlia. And then pectin, it is a component of the middle lamella of the plant cell wall. Hemicellulose is a component of pulp cell wall. And finally, chitin. Chitin is a component of fungal cell wall. So it is in the fungal cell wall and exoskeleton of arthropods. This is like frequently asked questions. So please remember about the chitin and its special property and its function. Okay, now let's go through the sentences. Pectin. Pectin is formed by galacturonic acid. Yes, it's correct, right? So here you see pectin is formed by galacturonic acid. Next, chitin is a nitrogen containing polysaccharide. Yes, so chitin is a nitrogen containing polysaccharide. Then amylopectin is a branch polysaccharide. You know, in starch there are two components, amylose and amylopectin. So amylose it is like a linear chain, right? So it is a linear chain, but amylopectin is branched. So this statement is also correct. Amylopectin is a branched polysaccharide. Next, glucosamine is the monomer of the hemicellulose. Okay, here is the problem. So glucose is not the monomer of hemicellulose, but pentose is the monomer of hemicellulose. Or you can say glucose is the monomer of chitin. It can be either chitin. Okay, so this is why the fourth statement is false. So it is the incorrect statement. And finally, inulin stores fructose as an energy store. Yes, that is correct. It as an energy source in the fructose because fructose is stored in the tubers of Dahlia where you can use it as the energy source. So this is about the polysaccharide. Okay, so the next question, let's move to the next question. It is about the cell junctions. So cell junctions, so what are cell junctions actually? They are the structures at which neighboring plasma membranes are joined. So they can interact and communicate via the sites of direct physical contacts. Okay, so these are situated at that point. What they do is they connect the internal chemical environment of the adjacent cells. It will connect it. So let's see. Correct about cell junctions. Connect the cytoplasm of adjacent cells. So that is correct, right? Why? Because cell junctions are the structures at which cytoplasm of the adjoining cells are joined. So of course, the cytoplasm of the adjoining cells are joined. So that is where the cell junctions are located. So that statement is correct. Next, tight. So you know there are three types of cell junctions. What are they? In the animal cells, there are three types of tight cell junctions. Tight junctions, then desmosome or anchoring junction, finally gap junctions or communicating junctions. So these are the three types of junctions. Now the second statement is tight junctions are found between muscle cells. This is false, right? Are they found between the muscle cells? No, the tight junctions are found in the skin epithelium. So it is found in skin epithelium. Okay, so that is false. So this is false. Next, let's see. So what are tight junctions? They will connect the plasma membrane of the adjacent cells and they are tightly bound by a specific protein, right? Which will form a continuous seal around the cells, okay? It will prevent the leakage of the extracellular fluid through the intercellular space. So that is the function of the tight junction. Next, gap junctions. Now what is the statement tells? Gap junction consists of special membrane proteins that surround the pore. Yes, that is correct, right? So now gap junctions also known as the Gap junction consists of a special membrane protein that surround the pore. That is that is correct. Now if you see the gap junction, they consist of special membrane proteins. Okay, they have special membrane protein that will surround the pore. where the iron, sugar, amino acids, they can pass through it. Okay, so this statement is also correct. So that is why I told there are quite a few mistakes here. So let's correct them. So those two statements are correct. Okay, so there is not an exactly one answer there. You won't get such questions in your final paper. Here we will just try to remove all the confusions. Okay, so gap junctions, they consist of a special membrane proteins that will surround the pore, which will allow iron, sugar, amino acids. Okay, and also they will allow the signal and material exchange between the cells through the direct connections. Okay, next, desmosomes connect the plasma membranes of adjacent cells tightly bound by specific proteins. Okay, this is false, right? Why? Because desmosomes, they mechanically attach the cytoskeleton of the adjoining cells by intermediate filaments. So there is nothing like plasma membranes or anything. It is attaching the cytoskeletons of the adjoining cells. So plasma membrane is related to the tight junction. The tight junction will connect the plasma membranes. So if they have given here tight junction, then the statement might be correct. But here they have told you desmosomes connect the plasma membrane and it is tightly bound by the specific protein. It's a false statement. And finally, anchor junction. Anchor junctions are also known as the desmosomes. They will mechanically attach the cytoskeletons of adjoining cells by microtubules, not by microtubules, right? It attaches the cytoskeleton of the cells by intermediate filaments. Intermediate filament. That is why it is also false. So this is about the cell junction. Cell junction might appear in your essay paper too. So still there's no question on essay question on cell junction, but it can be there. So it's better if you prepare for cell junction. When you write an essay on cell junction, write about the plasmodesmata also. Okay, it is not in the animal cell, but it is in the plant cell wall, but still you should include them under the cell junction. So when you write an essay, please don't forget to write about the plasmodesmata. Just write few sentences about that one also. Okay, now let's move to the third question. Select the phase of the cell division in the Tritoscandia pollen sac. So this is an important point here because that tells you if it is mitosis or meiosis. Okay, with the following features. Now, pollen sac, the cell division of the pollen sac means it will be meiosis, right? So it will be meiosis. That is how the gametes are formed. So meiosis is the process of the cell division where the gametes are formed. You see, this is, I think, totally from your resource book. Okay, chromosomes are attached with microtubules via centromeres. Okay, let's keep that statement. Let's go to the second statement. Chromosomes are arranged on the metaphase plate. Now here you have a point here, right? Metaphase plate, then it should be metaphase. So you know it is meiosis, you know it is metaphase. Now you should know, is it metaphase 1 or metaphase 2? Okay, you know it is metaphase 1 or metaphase 2. So if you see in metaphase 1, they might have mentioned homologous chromosomes. They might have mentioned now if you see this sentence, chromosomes are attached with microtubule via centromeres. If it is meiosis 1, they might have told you both the chromatids of the homolog are attached to the microtubules. But here they didn't tell anything like that. They just told you chromosomes are attached with microtubules via centromere. That confirms this is meiosis 2. So this phase is metaphase 2 of meiosis. Metaphase 2 of meiosis. So this is the way you can find this one. Okay, so next we'll see fifth question. Fifth question is about incorrect about the ethyl alcohol fermentation. So this comes, so ethyl alcohol fermentation, you should know few things. What happens here? So, you know, like in the aerobic respiration, the first step of the alcohol fermentation is also glycolysis, okay? So, the glucose will go through glycolysis process, and the pyruvates will be formed. So from one glucose, you will have two pyruvates. So what happens? These pyruvates will then, now here from pyruvate, it will turn to, from the glycolysis, the glucose will be turned to pyruvate. Now this pyruvate will involve into two steps. First step, it will be converted into acetaldehyde. Acetaldehyde by reducing the NADHs. Sorry, by releasing a carbon dioxide molecule. Okay, so the pyruvate will be reduced to will be converted to acetaldehyde by releasing carbon dioxide molecule. Then acetaldehyde will be reduced to ethanol using NADH. It will use NADH and it will be reduced. Okay, so this NADH is produced in the glycolysis process. It will be produced in the glycolysis process and it will be used here. So this is the process of ethyl alcohol fermentation. So the final hydrogen acceptor will be acetaldehyde, which is an organic compound. Okay, so most of the bacteria will carry out this ethyl alcohol fermentation, and the most common organism is the yeast, you know, right? So we use yeast in the bakery products where there will be ethyl alcohol fermentation and the carbon dioxide will be released out. So this is about the ethyl alcohol fermentation. Now let's see the statement. They ask for incorrect statement. One glucose produces two ethanol molecules and two ATP. That is correct, right? Because from one glucose, two pyruvate, so carbon dioxide, two acetaldehyde, two NADH, two ethanol. So two ethanols and two ATP is, two molecules of ATP is correct. Next, acetaldehyde was reduced into ethanol using NADH. That is right. Correct, right? Acetaldehyde turns to ethanol by using NADH. So that is also correct. Next, the final hydrogen acceptor is acetaldehyde. That's what I told you now. So it is the acetaldehyde is the final hydrogen acceptor. So that is also correct. Then most of the bacteria do this. That is also correct. And one carbon dioxide is released when one pyruvate is converted into one acetaldehyde. So one pyruvate, when converted to one acetaldehyde, it will release one carbon dioxide. That is also correct. So therefore, there is no answer in this question because all the 5 statements are correct. Okay, so that is about the ethyl alcohol fermentation. So there is lactic acid fermentation too. So please go through it also. So the final hydrogen acceptor will be an organic compound there also. Right. Now let's see the next question, which is the final question of mine. Incorrect about enzymatic reaction. Okay. Let's see. Now that is also incorrect statement. First one, enzyme bind with substrate and form a complex. That's correct, right? So it will enzyme will bind with the substrate and form enzyme substrate complex. So this complex will be then turned into enzyme product, enzyme and product. Okay, so that is correct. Next, right. Next, the shape of the enzyme determines its specificity. That is also correct, right? So the shape of the enzyme will tell how specific it is. Now in your book, in your resource book also it is mentioned. They have said the specificity of an enzyme results from its shape. That is why the reaction catalyzed by each enzyme is very specific. So that is also correct. Next, enzymes are not rigid structures. Yeah, they are not rigid, right? The interaction between the substrate and active site may change slightly, change the shape of the active site. So when not always the enzyme is complementary to its substrate. So during those times, the enzymes are not rigid structures. So they can change a little bit of the shape of the active site so that the substrate and active site become complementary to each other. So this is what we call it as induced fit mechanism, right? So induced fit mechanism. Okay, next, so that is correct. Then the tight fit not only brings the substrate molecules in the active site close to each other, but also ensures the correct orientation of the molecules. That is also correct. So due to this, it will help the reaction to proceed and catalyzes the conversion of substrate to product. So this is the reason for that. So that is also correct. Finally, catalytic action of the enzymes take place when enzymes collide with the substrate. That is also correct. So there is no answer for this question also, because all 5 statements are correct. So that is all for my part. Do you have